center of knowledge, innovation, history, culture, and biodiversity. This is the University of the Philippines, Los Baños. UPLB found its home at the foot of the legendary Mount Makiling in Los Baños, Laguna, a short distance from Laguna de Bay, the country's largest lake. It has its beginnings in 1909 as the UP College of Agriculture, a tent school for a handful of students. A year after, the Forest School, or what was to become the College of Forestry, also set up its home in Los Baños. In 1972, the UP College of Agriculture and the College of Forestry, that had by then become renowned institutions, were granted full autonomy as the University of the Philippines, Los Baños. Instruction, research, and extension in agriculture and forestry in the country took root and flourished in UPLB. Through its strength in these two fields, UPLB established niches in various areas: biotechnology, engineering, veterinary medicine, natural resources conservation, environmental science and management, human ecology, food science, economics and management, public affairs, and development communication. It drew its strength in the arts and other fields of science, from harnessing its expertise and human resource in the biological, physical, and social sciences. Today, UPLB degree programs continue to dominate over its counterparts. Over the years, UPLB has developed graduate programs that continue to attract students far and wide, including collaborative programs with international universities in food security, biodiversity conservation, and development. To make graduate programs more accessible to professionals, UPLB offers them off-campus. Recently, UPLB established the UP Professional School for Agriculture and the Environment in Mindanao, the country's food basket, in order to help build the knowledge capital that the country needs to attain food security. Instruction in UPLB is enhanced by a strong and vigorous engagement in research and public service that enables it to produce knowledge and generate technologies and innovations that promote sustainable living, making it a strong force in the war against hunger and poverty in the country. Extension and public service are the mechanisms by which UPLB brings this knowledge and technologies to the people, as well as for it to know what they need and to help shape policies in the country. UPLB's reputation as a graduate and research university was formed through the continuous aspiration for excellence and the mutual reinforcement between instruction, research, and public service and extension. It brings in partnerships with local and international universities and provides mobility to faculty members, researchers and scientists, and students, and enhances their competitiveness. Instruction, research, and extension and public service in UPLB reflect the dynamism, honor, and excellence that its forebears set as a standard for its leaders to aspire for.
The world is in a constant state of change now more than ever. It's a world that requires resilience, flexibility, and innovation to overcome challenges and to pursue growth. That is why the University of the Philippines, Los Baños is pursuing future-proofing initiatives to provide agile leadership and develop local and global communities amidst shocks and disruptions in an ever-changing world. guided by compassion, an openness to working together, and a daring to seek out breakthroughs, UPLB constantly pushes for academic excellence, produces relevant research, establishes academic and industry partnerships, and cultivates creativity, critical thinking, and innovation. To keep up with the fast-changing world and to strive for inclusivity in development, UPLB seeks to level the playing field and expand avenues for acquiring new knowledge. It is reshaping the learning experience, enabling students and educators to continue with the learning process in vastly different learning conditions. It is expanding its academic programs and supporting initiatives to create a conducive learning environment. To address the growing complexities of modern problems, UPLB continues to break barriers across disciplines to produce cutting-edge innovations and solutions that serve the needs of various communities. It is taking on challenges in food security and sustainability and coordinating research efforts to direct the university's resources toward critical research areas, making sure that the university-generated knowledge can reach its intended stakeholders and be applied to real-life contexts. UPLB is dedicated to its commitment to contribute to public good and social welfare as it continues to work with the local government and the community to address their most pressing needs. Amidst its engagements, UPLB remains as one of the top regional and global universities and continues to be recognized in the country and across the world. As the national university, UPLB aims to be a steadfast partner in development, as a producer of new approaches that minimize the cost and impacts of future shocks, and by honing leaders committed to knowledge creation, innovation and cutting-edge research, and public service. This is UPLB's roadmap to be an impactful institution in a constantly changing landscape. Responsive, innovative, future-proof.
A 112 years ago, the UP Board of Regents had a vision to develop their true firstborn college that will spearhead national development through agriculture. At the foot of Mount Makiling, Edwin B. Copeland, together with 7 faculty and 12 students, embarked on the journey to create the UP College of Agriculture, or UPCA. Through its 112 years of existence, the college has produced over 1,000 technologies and variety of plants and animals, 9 national scientists, over 30 outstanding young scientists, and more than 18,000 graduates contributing to agricultural development. UPCA, now College of Agriculture and Food Science, or CAFS, has been continuously contributing to nation building through its instruction, research, and extension function. A mere 10 years after establishment, the college was able to start breeding works with the production of sugarcane varieties with high productivity and resistance to Fiji and mosaic disease. In the 1920s, the importance of agriculture was intensified and UPCA was recognized as neighboring countries began sending their students to Los Baños because of the high quality of education. In the 1930s, breeds with superior characteristics such as the Burkhala pig, Los Baños Cantonese chicken, and filament cattle were developed. The 1960s was a fruitful decade for research. Makapuno, a coconut cultivar often made into a dessert, was pioneered and perfected. Meanwhile, the Dairy Training and Research Institute was the first in the country to freeze buffalo semen, paving the way for artificial insemination of local carabaos. Along with the establishment of UPLB in the 70s, the college received the prestigious Ramon Magsaysay Award for International Understanding, Asia's counterpart for the Nobel Peace Prize. In the same decade, the C4 plants were developed. The soil test kits were produced in support of the Masagana 99 program, and the mango induction technique was introduced. Continuing the production of quality research outputs in the 1980s, techniques to minimize post-harvest losses, the accelerated fish sauce manufacturing process, and the rapid test kit for the detection of pesticide residue on vegetables were developed. The 1990s saw many new varieties across species or crop types, including Sinta papaya, the first Philippine-bred hybrid, UPLB Gold, a durian variety, and Doña Amelita, a musaenda named after the wife of former President Fidel Ramos. The award-winning National Corn-Based Farmer Scientist RDE Training Program, or FSTP, was also established around this time and still remains as one of the university's primary extension programs. In the field of instruction, the book titled Integrating Sustainable Agriculture in the BSA Curriculum, the UPLB College of Agriculture Experience, was published in 1999. At present, the College of Agriculture and Food Science, or CAFS, continues to be the premier institution of higher learning in agriculture and food science, and remains committed to nation building through its instruction, research, and extension programs. In order to produce the best graduates, all of our courses are outcomes-based education, and continuous improvement of the curriculum is being practiced to meet the challenges of the 21st century. As testament of excellence, CAFS is constantly recognized by different organizations. In research, we continue to implement relevant studies with focus on the 12 priority commodities to address food security. Moreover, technologies are constantly developed to aid in responsible food production. Supplementing these are extension activities to disseminate the knowledge generated to improve food productivity and livelihood of the people. We believe that present technologies will future-proof our food systems. Some of our notable developments are iDetect, Coraginam, Plant Health and Pest Clinic, Pest Management Tools and Techniques, Smarter Pest Identification Technology or SPITech, Snap Hydroponics, Drone Technology, Rapid Immunofilter Assay for Virus Indexing, Nanofertilizers Fertigrow, Corbogrow, Nanobiosensors, Biochar. We at the College of Agriculture and Food Science will continue to promote and nurture a culture with innovative yet conscientious minds and cutting-edge skills to lead and serve the country and the region. We are one college with one goal towards sustainable food systems.
issues: pest invasions and outbreaks, crops diseases, massive use of pesticides, food security policies, confusing field practices. While Philippine agriculture has been a major arm of the government in achieving its long-term goal of food sufficiency, its objectives can only be attained fully in partnership with a strong pest management institution. The answer: the National Crop Protection Center. The National Crop Protection Center. The remnants of Rodent Research Center in 1968 paved the way for the establishment of the National Crop Protection Center on May 19, 1976, by virtue of Presidential Decree 936. Its founder, Dr. Fernando F. Sanchez Sr., exerted all efforts in expanding the rodent studies into more comprehensive pest management programs by forming teams of experts in rice, corn, vegetables, pest surveillance and forecasting, plant quarantine, pesticide chemistry and toxicology, and training and extension. Thus, the center's existence has been strengthened to provide solutions to the country's pest problems by developing, adopting and extending appropriate pest management technologies to farmers and formulating appropriate recommendations for use by government policymakers. An advisory board spearheaded by the Department of Agriculture served as guide in pursuing the goals of the center. After 12 years of clustering, the UP Board of Regents approved the restructuring of the College of Agriculture, renamed it as College of Agriculture and Food Science, and dissolved all clustered units and institutes to form independent units, again including NCPC. Today, the center rises up as a center of excellence in pest management, with its new leadership and experts that would continue the works and realize the vision of world-class excellence in crop protection and pest management. The center stands alongside premier UPLB institutions at the Agricultural Science Park of the University of the Philippines, Los Baños. NCPC is headed by a director, deputy director, and division heads, supported by a number of research and extension staff, and backed up by administrative personnel, unified by the standing goal to support agriculture programs to ensure food security, environmental sustainability, and the upliftment of the general public's quality of life. The staff work together harmoniously with each other and devotedly pursuing productivity. Researchers occupy laboratories with cutting-edge equipment to support pest management researches in the fields of plant biosecurity, pest management, pesticide management, and public service. Extension services are also active components of public service. Technical services are focused on Plant Health Clinic, the Quick Response Team, wherein NCPC addresses serious pest problems through quick response to pest outbreaks. The center also caters to pesticide residue analysis and pest database management. NCPC leads in national research and development programs with solid international and local partnerships, regional crop protection centers, Department of Agriculture regional field offices, Bureau of Plant Industry. The center's advisory board members that continue to provide guidance in the performance of its RDE functions are respected and well-accomplished institutions. NCPC strengthened and dynamic researches, human resources, and multi-institutional collaboration continue to meet the needs of various stakeholders, including the academe, LGUs, farmers, policymakers, and other government institutions. Welcome to the National Crop Protection Center.
Audio Jungle.
Good morning, everyone. I hope you're all doing fine. And now we're back to the NCPC Seminar Series 2022. We appreciate all the participants for joining with us on this new webinar topic, Nanobiotechnology Applications for the Management of Crop Pests and Diseases. I am Sarah Jean Manaday from the National Crop Protection Center, and I will be your MC for today's webinar. Before we continue, may I remind all the Zoom participants to keep your microphones on mute during the webinar. If you have questions or clarifications, please use the Zoom chat box. And for those who are watching from Facebook and YouTube, please write your questions in the comment section, and then our technical staff will gather your questions and then will be raised during the open forum. For those who are requesting for the e-certificate, kindly answer the evaluation form. The link will be found in the Zoom chat box or the comment sections of Facebook and YouTube. To formally start our webinar this morning, may I call on the NCPC Director, Dr. Barbara L. Quilly, to give us the opening remarks.
Magandang umaga sa inyo lahat.
Good morning, ma'am.
Good morning. So, to our resource speakers for today's webinar, Dr. Livia M. Fernando Corpus, Associate Dean for Research and Extension at the UPLB College of Agriculture and Food Science, and Associate Professor at the Institute of Crop Science, and Mr. Chris Lord P. Santos, Science Research Specialist at the Center for Natural Sciences and Environmental Research, De La Salle University. To our colleagues at the University of the Philippines, Los Baños, and to all our participants joining us virtually from different provinces in the country, ladies and gentlemen, a pleasant day to all of you. There is a continuous effort to look for new and innovative ways to manage insect pests and diseases in crops and to help farmers increase their yield. This is necessary since we are experiencing and we will continue to experience shifts in plant pest trends, primarily due to climate change and global trade. Among the novel approaches to crop protection is nanobiotechnology. So this is currently being used in medical science, in food technology, among others. So today, we will learn from the experts about how we can use nanobiotechnology in crop pest and disease management. Specifically, we will hear discussions on nanobiopesticides and nanobiosensors for plant pathogen detection. It is my hope that through this webinar, we will realize the great potential of nanobiotechnology. I also hope that more students, researchers, and scientists will be inspired to explore this field, particularly its prospects in crop protection. So with that, I wish everyone an insightful and meaningful webinar. Maraming salamat po, Hiraya Manawari.
Thank you, Doc Bambi. So before we hear our first speaker for today, may I invite everyone for the photo op. Ms. Charlotte will facilitate the photo op. Ms. Char?
Hello, good morning po. Yan. So makikibukas po ng ating mga camera for the photo op. Okay, so first panel po. 1, 2, 3, smile. Second panel po. 1, 2, 3, smile. Third. Okay, 1, 2, 3, smile. And last one po. 1, 2, 3, smile. Okay, okay na po. Thank you very much.
Yeah, so thank you, Ms. Char. Okay, so we will now proceed with our speakers. Our first speaker is a biochemist whose research is on the utilization of secondary metabolites from plant growth promoting bacteria for plant production and propagation. She works on the synthesis, characterization, and utilization of biocompatible nanomaterials for improvement of crop productivity, along with safety studies of nanomaterials in plants and microorganisms. Her research team also develops nanobiosensors for agricultural, environmental, and food safety applications and medical diagnostics. She is an associate professor at the Institute of Crop Science and an affiliate professor at the Institute of Chemistry and the current associate dean for research and extension of the College of Agriculture and Food Science, UPLB. She obtained her PhD in biochemistry, MS in Biochemistry, and BS in Chemistry from UP Los Baños. A DOS scholar and recipient of the Overseas Sandwich Program, she conducted her PhD research at Michigan State University. She had her postdoctoral studies at the Massachusetts Institute of Technology. Everyone, let us all welcome Dr. Lilia M. Fernando. Dr. Lilia.
Hello, good morning. Thank you for the kind introduction. Can I share screen now? Ayan, pwede na. Okay, so before I start, I'd like to thank NCPC through their director, Dr. Barbara Kawili, and all the organizers of the webinar series. Congratulations din po sa inyong webinar series. Okay, so as mentioned, our lab works on nanobiosensors. So for today's presentation, I'll be focusing on our work on plant pathogen detection. Okay, so according to the FAO, annually between 20 to 40% of global crop production are lost due to pests. And imagine, $220 billion. Well, ano pa to, hindi pa yata to updated. Ang nawawala due to plant diseases and $70 billion naman globally due to invasive insects. And due to increased trade and travel, lalo ngayon na open na ang ating borders, may risk ulit for plant pests spreading into new areas across borders. So we had precautions before sa human because of the COVID-19, but sometimes we overlook on how we can protect our plants and animals as well. So nagbibigay talaga ito ng threat to the well-being of our plants and livestock. If you remember, baka yung iba sa ating audience ngayon, hindi naman siguro. When our country, lalo na dito sa ating area, was devastated of the coco lysap. We're glad na solusyonan naman po ito. So there's really a need for us to be proactive in terms of biosurveillance. And we're glad that our government, through the DOST, the Department of Agriculture, is strengthening the biosurveillance and biosafety aspect of this particular sector, agriculture. So our team focuses on solutions utilizing nanotechnology. So briefly, nanotechnology is the understanding and control of matter at dimensions between 1 and 1,000 nanometers. However, they became strict in terms of definition. So now, nanotechnology mostly makes use of 1 to 100 nanometer size of particles. So imagine how small these particles are, 10 to the minus 9 meter. So it's a convergence of chemistry, physics, engineering, and biology applied in health, agriculture, and other sectors. So the idea on nanotechnology came from Dr. Richard Feynman, a Nobel Prize in Physics, where he mentioned that what I cannot create, I do not understand. So according to him, just like the living cell na maliit, pero kaya niyang mag-work on its own. So according to him, he can also engineer a small particle such that this particle can grow and, maneuver and can do a lot of things. So that was an idea before, but then the term nanotechnology was coined by a Japanese professor, Prof. Norio Taniguchi. Nanotechnology was used initially in the semiconductor industry, and eventually it was utilized by other sectors, especially medicine and now agriculture. So just to give you a perspective, these pennies, 1 centimeter, so 10 to the minus 2 meter, yung ating strand ng hair, 10 to the minus 4, viruses, about 1 micrometer. So estimate lang po ito. And gold nanoparticle that we make use in medicine and agriculture, about 10 nanometers. So sometimes 20, 50 nanometers, depends on the application. So for characterization, we use electron microscopes for nanomaterials. Specifically for the nanosensors, we use metallic nanoparticles like gold and iron oxide nanoparticles. With regards to pathogen detection, there are various methods that's commonly used. One is plating, which is the conventional method. And here are some parameters to compare the diagnostic methods. So the assay duration, detection limit, specificity, on-field portability, and sensitivity. So plating, then we go down to immunological, such as ELISA techniques. Then nucleic acid, such as PCR-based technologies. And lastly, nanotechnology-based techniques. So among the four, we have a promise in using nanotechnology-based techniques because of the short duration or rapid assay time. Detection limit is in the femto level, so 10 to the minus 15, yung pwede niyang i-detect. And it has excellent specificity, sensitivity, and portability. Okay, so to summarize, here are the advantages why we prefer to work on nanobiosensors. So the last bullet here emphasizes its compatibility with data processing technologies. Okay, so our work started on nanobiosensors through Michigan State. We have our balik scientist awardee before, Dr. Vanchi Alusilha. So we work since 2007. So almost 15 years na po yung aming trust sa bawat isa, and exciting po ang mag-collaborate with them. So we maintain the relationship, and we're glad that a lot of students and colleagues were able to train in her laboratory. So the Nanobiosensors program that we had started way back when I was with UPLB Biotech. We started with the detection of food and waterborne pathogens and baka and banana viruses. Okay? And I also worked on tropical diseases such as dengue and JEV, but focusing on mosquito because UP Manila naman po ay sa clinical sample. So mostly biosurveillance yung atin pong purpose on nanobiosensor technologies. Okay, so how do we work on this on the nanobiosensor program? So first we have the triage technique. Para pong pag nagpa-consult tayo sa mga clinics or hospitals, hindi naman nila agad ititest yung sample natin. Or kapag i-check mo na may lagnat ka ba, ganyan. So tayo, since halimbawa may mga field samples tayo, sayang naman kung ipapasok at natin agad sa diagnostic or DNA-based technique na wala naman palang probable disease dun sa ating sample. So for the triage technique, we use magnetic nanoparticles. So later on, I'll be showing a bigger figure. So this relies on matting or spreading of the magnetic nanoparticles. So when we have the pathogen, we're not specific yet at this stage. If we have the pathogen, kakaroon ng matting or spreading yung ating nanoparticles. If wala, so parang ganito lang po siya. Naka-focus lang or concentrate dun sa end ng ating tube. Okay, so only those that had the matting will be subjected to the DNA-based or more specific detection method. So as mentioned earlier, we want this to be a biosurveillance system. So eventually, we want to have an app in our computers that can notify areas. Halimbawa, yung example ko na po yung aming first study on cacao. So halimbawa sa Mindanao, may na-detect na tayo, then we can notify other areas para ma-contain muna yung ating mga seedlings, not to be transported to other areas of the country. So that's the dream. So we started on cacao. So how did we start with our favorite, kasi chocolate? Through Dr. Divina Amalin. So she's also an alumnus of UPLB. So she tapped me to work with their team so we can have a detection system for the causative agents for cacao diseases. Okay, so for instance, the black pod rot, it accounts for up to 70 to 80% annual cacao production loss. Okay, so we want to prevent this, yung mga black pod rot. So the idea of nanobiosensors is to detect the pathogen before pa po, So wala pang symptoms yung halaman, dapat makita na natin kung meron na nung pathogen. So just like the previous table that I mentioned earlier, I showed earlier, the following diagnostic methods are used for the detection of the causative agent of black pod rot, Phytophthora palmivora, so culture method, PCR, and ELISA. So we want the detection method na pwedeng gawin sa field. That's why we work on a nanobiosensor system as an alternative for these detection methods. So we don't want to replace this, Nandyan lang po yan. We're also working on some validations. So our nanobiosensors can be used in the farm. Yun po talaga yung ating purpose for this technology. So as an example, for the detection of Phytophthora palmivora, and later on, I'll be incorporating slides for Lasiodiplodia theobromei, the causative pathogen for vascular streak disease. So for the screening or the triage technique, as mentioned earlier, sorry po medyo malabo. Dapat may spreading kapag may Phytophthora palmivora. This technique was previously used in the detection of clinical samples, dengue, tuberculosis. So sabi namin, why not use or try in plant pathogens? So yun po, it relies on some chemistry po sa surface nung pathogen and that of the magnetic nanoparticle. So we're also working on image analysis so we can quantify the area or the matting of those that are positive for this stage of the diagnosis. Okay, so for the DNA-based detection, we have two methods. One is the colorimetric detection, and the other one, electrochemical detection. So I'll first focus on colorimetric detection.
Yung mga natanda, bilis.
Okay, so colorimetric detection, this is one of the techniques or principles that we adopted in our nanobiosensor. So we use gold nanoparticles. So dito po sa left, we have the red circle as the gold nanoparticles, and on the surface are the oligonucleotide probes. So these probes were designed and are complementary to a region of the DNA ng ating pathogen. So dito sa taas, when we have the target, it should be complementary and be able to hybridize with the oligoprobes. And with the property of gold nanoparticles having this these arms, because of the hybridization of the target with the oligoprobe, so disperse sila, hence there's no aggregation. We obtain the pink or red to pink color of our gold nanoparticle. However, when we have the non-target, since no hybridization will occur with the oligoprobe that we designed, mag-aggregate po kasi hindi na stable yung arm ng ating gold nanoparticles. So we usually obtain purple to clear color of our solution. So how do we do this in the field? Okay, so we have our tube with the target DNA. We're working on an on-site DNA extraction technique. So hopefully we can validate this in the field already, yung on-site DNA extraction. Okay, so target DNA, then we add the oligoprobe, perform hybridization, and then add gold nanoparticles. Okay, and add salt, just to have some ionic stabilization in our solution. And eventually, with the target DNA, we obtain the pink color as mentioned earlier. How about with the non-target? So again, we start with our sample, yung non-target DNA. So syempre, we don't know if it's really the target or the non-target. So just an illustration po itong ating slide. Okay. So we obtain the purple or purple to clear color. Nag-aggregate po yung ating gold nanoparticle. So we work on optimization of various parameters in order to arrive with that technique on colorimetric detection. So as an example, kanina po yung Phytophthora, now I'm showing you that of L-theobromate. So this is the blank. Sorry po, purple dapat ito, medyo pinkish yata dito sa ating photo. Purple to clear yung nagiging color nito. And with L-theobromate, it's pink. Okay. And here's another data. This is the blank and L-theobromate. And we tried using Phytophthora palmivora. Okay, and a combination of L-theobromate and P. palmivora. Still, the colorimetric method was able to detect L-theobromate. So the oligoprobe is specific to L-theobromate. That's why it was not able to detect Phytophthora palmivora. So just a quantitative representation of the previous slide. So we have a lower absorbance reading because of the pink color of the gold nanoparticle. So no aggregation was observed with these two. Yung L-theobromate po dito sa bar na to and the last bar on the right. Okay, how about the electrochemical detection? Okay, so we make use of a potentiostat in order to quantify the sample in our the sample or the DNA that we have or the target that we obtained from the field. Okay, so yung medyo matagal lang po dito yung ating sample preparation, but the electrochemical detection will take about 30 seconds. So this is battery operated. Ang kagandahan po nito, I mentioned earlier, we also detect foodborne and waterborne pathogens. So in calamity affected area, malaking tulong po ito. And now we're using it in plant pathogens. Okay, so we just use about 50 to 100 microliters of the sample. So yung DNA po natin through on-site extraction, actually isang drop lang niya dito sa ating screen printed carbon electrode, and then we check the current response. Okay, so just an illustration on how this works. One of our protocols before is to use a PANI or polyaniline coated magnetic nanoparticle. And this is the oligoprobe, yung kanyang tail. And we have another oligoprobe here. So we have oligoprobe 1, yung ating black, and oligoprobe 2, yung ating blue tail dito. Now, the blue circle is biotin. So later on, makita po natin on bakit kailangan dalawang probes, and why the need for biotin and magnetic nanoparticle or PANI. So if we have our target DNA, magha-hybridize po siya. Lumampas. Anyway, ano po yan? Dapat mag-hybridize to, hindi lang po maayos sa animation. Supposedly, it should look like this. So by virtue of the magnetic nanoparticle in the field, we don't use centrifuge anymore. We just use a magnet to concentrate our sample. So only those with the target DNA, because of this oligoprobe na nag-hybridize po sa part na to ng ating target, will be left in our solution during washings. Okay, so from that, we use the screen-printed carbon electrode with embedded gold nanoparticles just to intensify the electrochemical signal. And why the use of biotin? Dahil maglalagay rin po tayo dito ng streptavidin. So there's an affinity, special affinity between biotin and streptavidin for them to be stable in this platform, yung screen-printed carbon electrode. And from that, we can detect yung signal using the potentiostat. So just an example of the voltammogram that we usually obtain. This is from magnetic nanoparticle, but I'll just show you when we optimized yung concentrations po ng ating sample for Phytophthora. So the green curve here is for 0.1 nanogram per microliter of the DNA. So it can detect up to 0.1, as low as 0.1 nanogram per microliter. Okay, and this is one of the selectivity studies that we conducted using the electrochemical nanobiosensor. So we can selectively detect Phytophthora palmivora if we have or among the other pathogens that we have in the sample. And the limit of detection, as mentioned po, it can go as low as 0.1 nanogram per microliter. So here's another data where we used pure culture of Phytophthora palmivora and artificially infected pod. Okay, so compared po dun sa healthy pod and yung naturally infected pod, hindi siya masyadong significant yung kanyang difference with the healthy pod. So we're working on some optimization studies here. Sometimes we need to do spiking as well to detect this. Okay, and just to show you yung initial studies po namin on the field validation of the lab and the field validation of this work. So again, the dream is to have this eventually, to have the handheld potentiostat attached to a packet PC and eventually have a database or a cloud, where we can have all those information. Hopefully, we can work with the DA and the BPI for the application and use of this nanobiosensor. So now we're working on lab and field validation of this nanobiosensor. So here's the nanobiosensor system that we have. And in relation po sa roadmap ng Picard, I checked last night, di pa nila na-update ng 2022. So mostly promotion and commercial application and validation po ng ating mga technologies like the nanobiosensors ang focus ngayon for the detection. Okay, so other applications of the nanobiosensors, we also use the same prototype in the detection of the following pathogens. So in banana, we have Fusarium and the bunchy top virus. In white potato, Phytophthora infestans, the potato virus Y, tomato, Ralstonia, mung bean viruses, peanut, we have Cercospora. So my question mark, because according to one of our study leaders, Dr. Fe de la Cueva, we have to check this, no? Hindi daw po ito yung common na organism or pathogen dun sa peanut sa region 1. So also, yung ating post-harvest processing ng cacao. So now we're working on the detection of the pathogens such as Aspergillus niger and Flavus, which produce ochratoxin and aflatoxin. So we're collaborating with cooperators in Davao and other areas here in Laguna who are working on, or farmers, cacao farmers working on the processing of cacao. So titignan po namin yung iba-ibang stage ng post-harvest processing para ma-check natin kung saan doon mataas yung population ng niger, Aspergillus niger, and Flavus. So for now, we focus on the pathogens. So same prototype as what we used in Phytophthora and Lasiodiplodia. So sa insects, halos ganun din po, ano, because we know that insects are vectors of pathogens. Okay, so we're working on some studies as well, collaboration with Dr. Amalin, to include insects in our research on nanobiosensors. So this is the final product that we want to have. We actually have a registered trademark now. We call this Alert Plant Pathogen Test Kit. So we have here the DNA extraction kit already and the colorimetric, and if needed by the customer, now we'll have the potentiostat as well. So hopefully we can complete validation studies up to next year and have this pre-commercialized already. Okay, so hopefully we can contribute to the SDGs, ano? Especially number 3 on good health and well-being and responsible consumption and production. So on behalf of our team, we would like to thank our funding agencies and collaborators. Salamat po ulit and magandang umaga.
Thank you, Dr. Leah, for the informative and very interesting presentation regarding nanobiosensors. So before we proceed,
so on behalf of our team, we would like to thank our funding agency and collaborators.
Okay, so before we proceed with our second speaker, we will have the open forum for Dr. Leah. And for the audience, if you have questions or clarifications with regards to her topic or presentation, this is the right time to ask. You can use the Zoom chat box or comment section in the Facebook or YouTube. To lead us in the open forum, may I invite, before we proceed with to lead us in the open forum, may I invite Mr. Jasper Sarmiento from NCPC to facilitate the question and answer portion.
Thank you, Ms. Sarah, and thank you to our speaker, Dr. Leah, for a very insightful lecture on nanobiotechnology. No? So very promising yung applications ng nanobiotechnology. So right now, we are currently monitoring the Zoom app and YouTube Live for questions po. No? So if you have questions, wag po kayong mahiyang magtanong sa or i-type yung questions po natin sa chat box and sa comment section. So siguro habang naghihintay po tayo, no, ako po muna yung magtanong. Dr. Leah, ano po kaya yung mga challenges pagdating po sa ganitong klase ng research?
Okay, thank you, Jasper. Very good question. Isang challenge yung specificity. That's one problem, or sabihin ko na challenge that we observed, especially sa aspergillus, dun sa post-harvest or mycotoxin project namin. Because they're of the same genus, so medyo mahirap kaming makakita ng magkaibang regions nila. Medyo tricky pa. So when we check Aspergillus niger, sometimes we can also detect flavus, yung Aspergillus flavus. So we're working on that. And sometimes may overlap pa, kasama namin yung Lasiodiplodia na di-detect din kahit DNA-based. So that's one aspect that we're working on, no, na yung specificity niya ay ma-improve pa. Yun, yung isang challenge namin. And also, when we work on field validation, mas marami pa na magiging challenge because we will be working on an on-site DNA extraction. So, halimbawa yung mga soil-borne pathogens natin like Ralstonia would be difficult for us to extract from the soil because of the siyempre mas iba yung matrix than just using culture in the lab. So yan yung mga challenge na aming kinakaharap sa lab, and it's exciting actually, no? Kaya nga sabi nila, research, you always search for solutions.
Yan, thank you po, Dr. Leah. So meron po tayong question sa Zoom. So from Emerson J. Ibacara. So ang tanong po niya is the technology utilized commercially?
Okay, not yet. No, we're still working on the validation of the technology, and hopefully by next year we can go to pre-commercialization. That's when we work with prospective industry partners to also test the product, and kailangan din ng mga laboratories, mga licensed or certified laboratories na natin that we can work on for the validation studies. So hindi pa po siya commercially available, but we're working on it para mabilis, no, yung ating process.
Thank you po. From Zoom uli po. So from Harold Paniza. So during the conduct of the activity, does it affected by environmental factors, for example, temperature? Also knowing na it needs Also knowing na it needs DNA extraction kit, it is still considered as DNA-based technology. Also, why gold metal nanoparticle? Medyo costly po kasi ang gold. Have you tried copper or silver po?
Okay. Ilan ang question ni Harold?
Ang dami po niyang question.
Okay, sige. Okay, thank you, Jasper. So the first question, if it's affected by the environment like temperature, yes, yung ating process, but compared to enzyme-based techniques, DNA is more stable sa temperature. That's why we focused on DNA. For the on-site extraction, we have actually hindi naman ganun ka-tricky, kasi parang yung usual extraction din sa laboratory, nagagamit tayo ng mataas sa temperature. So we're using actually nag-try na term, na may range lang ng temperature doon na pwede nating gamitin for the on-site extraction. So for the temperature, not much yung kanyang effect doon sa ating result. Okay? Lalo na we work on DNA. As I mentioned, it's more stable compared to enzymes or antibodies. To be antibodies na lang to be specific. The other question, na-mention ko na rin yung
daw po yata yung
okay. Well, we actually have initial cost analysis already. Yung triage technique natin na magnetic nanoparticle, about 10 pesos lang yon per sample. While the gold nanoparticle, compare dito dun sa ating conventional method like plating, minsan 500 to 1,000 yung ating binabayad doon for analysis. We only pay 50 pesos dun sa ating DNA-based na technique. The reason, gold is medyo expensive siya pero super konti, no? Konting-konti lang yung ating ginagamit for our assay. Okay, kaya hindi talaga siya sayang. Even when we consulted with our balik scientist, si Dr. Alusilha, they used it in dengue and tuberculosis before. Sabi nila it's super cheap compared to the PCR and the culture method. So why not use copper or silver? Because of the property of gold, we want to use it in colorimetric detection. And compared to copper and silver, mas pronounced yung property ng gold sa pag-change ng color at that particular size. So it's about the chemistry behind gold, copper, and silver nanoparticles. Actually, we have studies as well using copper and silver, but as hindi siya sa sensing, as antimicrobial agents. Pwedeng gamitin yung copper and silver. But for sensors, we use gold, mas stable din siya. And also, yung kanyang electrochemical response is more pronounced and mas applicable in our application compared to copper and silver.
Ayan. Another question po for Cordup, yeah. What are the advantages and disadvantages of using this technology po?
Okay. So yung rapid detection time, kasi we can detect our pathogen, lalo na yung DNA-based, in 1 to 2 hours. So yung matagal actually yung sample preparation. Okay, the on-site extraction ng DNA and other processing. And another is that it's more specific and more sensitive. Yung specificity nga lang, sabi ko kanina, kailangan pa namin siyang i-optimize, because there are some cross reactions. And it really happens. It really happens. One time, I talked to one of our collaborators, yung dengue. There are 4 serotypes. And nagko-cross-react. Well, it doesn't matter. It's still dengue. Ganon, di ba? Minsan yung ating perspective. So, with this, hopefully, we can, we're working on, may mga naisip na kaming steps para mas natin, ma-identify yung ating mga organisms. Mahirap yung cross-reaction. It's really challenging. Okay, I hope I was able to answer the question.
Okay. Another question po from Zoom kay Dennis Rocamora. So, good day po, Doc Lia. Inclusive lang po ba sa plants ang kagamitan ng nanobiosensors to detect diseases? Baka pwede rin po sa animals. Thank you po and God bless.
Okay, sige, thank you sa question. Hindi lang siya sa plants, we actually ang ASF, ang alam ko meron na ring work dito ang CLSU, for DNA-based nanobiosensor detection. And here in UPLB, we have a collaboration with Dr. Dong Angeles, our D3 director. So they're detecting salbutamol naman sa mga feed samples natin. Kasi yung salbutamol pala minsan ginagamit ng livestock industry para kumonti yung taba dun sa mga meat. Siyempre ayaw natin nung maraming taba. Unknowingly, meron palang salbutamol minsan na nilalagay para malesen yung fat. And we don't know yung effect nito sa health natin pag kinonsume natin yung meat. Kasi syempre may residual salbutamol pa doon. So that's another study that we're working on. So nanobiosensors can be applied to anything na gusto nating i-detect, as long as we know the principle that we want to use in the development ng sensor.
Another question po from Harold Paniza. So, kumusta po ang status sa efforts nyo with regards po sa banana disease causing pathogens? Yung TR4 and PBTV. Do we have an opportunity to collaborate about this po?
Okay, so very good question din kay Sir Harold. Taga saan po kayo? Baka yes, we can collaborate with you. Our project on FOC-TR4 and BBTV, natapos na po yung phase 1. We're working on a proposal for the validation po ng ating sensor for the FOC-TR4. Unfortunately, TR3 lang yung ating nagamit because during the lockdown, we were not allowed to go to Davao. Kasi we were not allowed to transport po yung TR4. So yes, we can work with you for this pathogens. Exciting po during the validation stage.
Okay. Ayun, last question na po yata yun. Ang sabi po ni Mr. Harold is taga Davao daw po siya.
Okay, thank you. Sige po.
So, ayun, muli po, salamat po Dr. Lia for the very informative lecture, and salamat din po sa pag-accept ng invitation, at salamat din po sa ating viewers for the participation po. So binabalik ko na po kay sa MC natin, Ms. Sarah.
Okay, so thank you, Mr. Jasper, for facilitating the open forum, and also to our guest speaker, Dr. Lia. We will now proceed to our second speaker. Now, our second speaker is a science research specialist at the Biological Control Research Unit of the Center for Natural Sciences and Environmental Research, De La Salle University. He works on the development of biomosquitocidal formulations from plants and bacteria against insect pests, and the use of semi-chemicals as trapping systems for insect pests. He is also a lecturer at the Department of Biology, College of Science in the same university. He previously worked in UPLB, teaching courses on organic and biochemistry at the Institute of Chemistry, College of Arts and Sciences, and working on the development of nanobiopesticide against Fusarium wilt in high-value crops at the National Institute of Molecular Biology and Biotechnology. He received his bachelor's degree in agricultural chemistry and his Master of Science degree in biochemistry from UP Los Baños, with full scholarship from the USTC and the USTP Cards Graduate Research Education and Assistantship for Technology Program. So everyone, let us welcome Mr. Chris Lord D. Santos.
Good morning, everyone. I hope you're all safe after the super typhoon. Thankfully, the weather is better today. We are able to meet for this seminar. So for this part of the seminar on nanobiotechnology applications for crop pest and disease management, I will be discussing in particular the application of nanobiopesticides. So the title of my lecture is Nanobiopesticides from Plant Growth Promoting Bacteria, or PGPB. So the content of this presentation is actually a portion of my master's thesis, which I conducted at Institute of Chemistry, UPLB, the National Institute of Molecular Biology and Biotechnology, UPLB, and the Institute of Plant Breeding. So this was supported by the UST and UST PICARD. Let me just turn off my video since my internet connection is a bit unstable. Yeah. So earlier, during the presentation of Ma'am Leah or Dr. Corpus, she discussed on the application of nanobiopesticide or nanobiosensor technology. So in particular, they utilize DNA-based nanobiosensors using specific nanoparticles like the magnetic nanoparticles and the gold nanoparticles in order to detect specific microbial pathogens. So what happens now after you detect the pathogen? So usually we do plant disease management. So we use control measures like cultural control, chemical control, and biological control, among others. And the type of control measures or control measure that you need to use usually depend on what stage of the disease cycle we are considering. So we might do preventive measures before pathogen infection or curative measures after the disease has already been established in the host plant. So in our group, we are concerned with biological control or biocontrol. So we use biocontrol agents in order to control the growth of specific microbial pathogens that are typically controlled using synthetic pesticides. But as we know, there is an increasing concern with the use of synthetic pesticides because they are generally more toxic and they can cause damage to the ecosystem and the environment. So instead, we use biocontrol measures that are generally environment friendly, have low cost and have low toxicity, and usually the development of resistance is slower. So biocontrol involves the use of living organisms or their products to eliminate or reduce the damages or losses due to the specific pest or disease. So in this study, I did not use a live organism, but instead a product. So in particular, I used an enzyme as a biocontrol agent. So enzymes, again, are proteins that catalyze specific chemical reactions in biological systems. And they are good biocontrol agents because they have high selectivity, they can be produced in large amounts, and they are present in many organisms. So in particular, in this study, we are concerned with the biocontrol of a specific fungal pathogen, which is Fusarium oxysporum. And Fusarium, like other fungi, has chitin and beta-1,3-glucans as the primary components of their cell wall. So this is the general structure of chitin. And chitin can be degraded, can be hydrolyzed by a specific enzyme called chitinase. So in this study, we utilize chitinase as a biocontrol agent against a fungal pathogen. However, chitinases and enzymes in general are sensitive to changes in pH and temperature, and they have low stability. So in order to solve this problem, we use nanotechnology or nanobiotechnology. So as mentioned by Ma'am Leah earlier, nanotech involves the use of materials that usually have sizes around 1 to 100 nanometers, around that size range. So the use of these nanomaterials usually results in higher stability, efficient delivery, and enhanced activity. So yung concern natin for the use of nanotechnology and in particular the nano-encapsulation technology is to make the enzyme more stable so that it can be applied in the field as a biopesticide. So for a brief summary of this study, so we produced enzyme-loaded nanoparticles, so specifically the chitinase enzyme. So first, we screen different PGPB, plant growth promoting bacteria, for the production of the chitinase enzyme. And the bacteria or the PGPB that showed positive result from the screening was used for chitinase extraction, and the purified chitinase enzyme was characterized and was used for encapsulation. So the enzyme-loaded nanoparticles after encapsulation was characterized. So their size and morphology was described, the stability and the in vitro antifungal activity using the Fusarium as the test pathogen. So for the first part, we have this screening. So for the screening for chitinase activity, I use a qualitative plate assay where I use colloidal chitin as the sole carbon and nitrogen source. So colloidal, excuse me. So I inoculated different PGPB, plant growth promoting bacteria in plates containing chitin. So the positive result for the screening is indicated by the presence of the zone of lysis around the inoculation site. So the zone of lysis indicates the production of the chitinase enzyme by the bacteria. So the bacteria or the PGPB that showed positive result was used for the production of the chitinase. Again, here we used colloidal chitin as the sole carbon and nitrogen source in order to induce the production of, or in order to stimulate the production of the chitinase enzyme. So after incubation, after submerged fermentation, the crude enzyme was extracted. So after extraction, the enzyme was purified and was used for the encapsulation. So in this study, I used two encapsulation methods, lipid encapsulation and carbohydrate encapsulation. So using two different wall materials. So for the lipid encapsulation, the materials that we used was soy lecithin, which is a phospholipid, so lecithin or phosphatidylcholine. So in this methodology, I used thin film hydration method in which the wall material or the lecithin was dissolved in ethanol, which was subsequently removed under reduced pressure using a rotary evaporator. So this produces a thin film of the lipid on the surface of the flask. Then the film was rehydrated using the enzyme solution, resulting in the formation of multilamellar liposome vesicles, which are larger in size. So in order to produce yung smaller nano-sized unilamellar liposomes, the solution was sonicated for a specific amount of time. So in order to produce the smaller nanoparticles. So for the other method, which would be the carbohydrate encapsulation, the nanoparticles were formed via a method called ionotropic gelation using chitosan and tripolyphosphate. So first, chitosan was dissolved in acetic acid, in order to increase its solubility in water. Then a specific amount of enzyme that is mixed with the tripolyphosphate was added. So the tripolyphosphate acts as a cross-linking agent in which the phosphate groups in the polyphosphate molecule form phosphoramide bonds with the amino groups of chitosan. So this results in the formation of the chitosan polyphosphate nanoparticles, and in the process, yung enzyme particles are immobilized or trapped in the structure of the nanoparticles. So the enzyme-loaded nanoparticles were again characterized. So their size, their particle morphology, their stability, and their antifungal activity were evaluated. So in terms of the particle size and morphology, so DLS analysis, results I forgot to show here, but DLS analysis showed that the particles have sizes around 100 nanometers, and this was confirmed through electron microscopy, so SEM analysis, which showed spherical particles with sizes around 100 nanometers. So in terms of stability, we wanted to know what would be the effect of the encapsulation in terms of the activity of the enzyme when it is subjected to extreme values of pH and temperature. So here we see that both lipid and carbohydrate nanoencapsulation actually made the enzyme more stable under acidic, so at pH 4, and at pH 9 or basic pH. Yeah. So in the free enzyme, we can see a significant decrease in activity at these extreme pH values. So at pH values away from the optimum pH of the enzyme. While for the encapsulated enzyme, the nanoparticles, the residual activity is still high even after 3 hours. So the stability of the encapsulated chitinase is also observed when they are subjected at high temperatures. So we looked at the change in the activities or the residual activities at 60 degrees, 60 degrees and 90 degrees. And compared with the activity of the free enzyme, here we saw that the encapsulated enzymes are much more stable, much more stable at these increased temperatures at 60 and 90 degrees Celsius. So these results show that encapsulation, both lipid and carbohydrate encapsulation, can significantly stabilize the enzyme, which is important if we will be using these nanoparticles as nanobiopesticides in the field, where we can where the conditions can be unfavorable for the enzyme. Yeah. So for the last part, I also evaluated the effects of nanoencapsulation on the antifungal activity of the chitinase enzyme in vitro. So we use 2 pharma species of Fusarium oxysporum. So these are Fusarium oxysporum commerinum and Fusarium oxysporum lycopersicei, causing Fusarium wilt in cucumber and tomato. So shown here are the plate cultures of the 2 fungal isolates, as well as the photomicrographs showing their conidiospores. So we evaluated the antifungal activity in terms of their capability to inhibit mycelial growth in plates amended with the specific treatment, the nanoparticles. So the results show that the chitinase nanoliposome, so this would be the enzyme encapsulated using the lipid wall material, the producing the nanoliposomes. So the nanoliposomes inhibited the mycelial growth of the 2 fungal pathogens better than the commercial biopesticide at 50 milligram per ml concentration. So this is compared with the positive control, the biopesticide Serenade, which contains live cells of Bacillus subtilis. So we use the recommended rate of the biopesticide. So results also showed that the encapsulated enzyme has a higher activity than the free enzyme and the wall material, because we included these in order to show that the activity is due to the enzyme and not the wall material, not which is yung phospholipid. Yeah. So for the chitosan nanoparticles, so the chitinase encapsulated with the carbohydrate wall material, at we saw complete inhibition of the mycelial growth at 50 milligram per ml concentration, and at 25 mg per ml, the nanoparticles inhibited mycelial growth. The mycelial growth inhibition of the nanoparticles at 25 mg per ml is comparable with that of the commercial biopesticide at the recommended rate. So the higher activity in terms of mycelial growth inhibition that is observed for the chitosan nanoparticles may be attributed to the inherent antifungal activity of chitosan, which was used as the wall material. So this results indicate the effectiveness of the encapsulated enzymes as biocontrol agents against this specific pathogens, Fusarium sesperum. But of course, the effectiveness of these materials, these nanoparticles, must still be evaluated using screenhouse and field bioassays. So now, how do we explain the apparent increase in the activity of the enzyme? So for the nanoliposomes, in general, there are several mechanisms that can use to explain the increased activity of the nanoliposomes. So first, we have the leaky liposomes theory, in which the core material, so whatever it is that is inside the nanoliposomes, so the core gets released from the liposomes near the cell wall of the fungi. So if it is the chitinase enzyme, so the release of the chitinase near the cell wall promotes a higher activity. So the second mechanism would be the collision mechanism, in which the core gets out of the liposomes when they collide with the fungal cell wall. So the collision results in the release of the core, also in this case the enzyme. So the third one would be the fusogenic mechanism, in which the phospholipid layer in the nanoliposome merges or fuses with the fungal cell, the fungal plasma membrane, resulting in the release of the enzyme inside the organism. So for the chitosan nanoparticles, the increased activity may be attributed also with the increased attachment of the nanoparticles to the fungi, to the fungal pathogen, which is due to the highly positive surface charge due to the presence of amino groups in the chitosan. So the highly positive nanoparticles interacts favorably with the usually negatively charged surface of the fungi. So this results in the release of the enzyme near the pathogen. So what are some future prospects of this technology? So in particular, the chitinase nanoparticles may be evaluated in terms of their effectivity as biocontrol agents against insect pests, since for insects, their exoskeleton is also made up of chitin. So we can explore the effectivity of the chitinase nanoparticles in controlling insect pests. Also, a more targeted application using metabolites that can control specific pathogens can also be explored. So other core materials other than enzymes may be used for a more specific control of specific pathogens. And third, the evaluation of the effects of the nanoparticles on non-target organisms must be evaluated in order to see the effects of these nanoparticles on the ecosystem, particularly in the rhizosphere. Where these nanoparticles will be applied. So that will be the end of my presentation, and I hope you learned something. So thank you, and again, good morning.
Okay. Okay, so thank you, Mr. Santos, for sharing with us your work and knowledge on nanopesticides. Now we will proceed to the open forum, and to lead us, may I invite again in the platform Mr. Jasper Sarmiento, our open forum moderator.
Yun. Salamat, Miss Sarah, and salamat to Mr. Chris Lord for that very interesting lecture on nanobiopesticides. So we are currently monitoring nga po yung YouTube Live and Facebook, Zoom app. So if you have questions po, you can type in your questions sa chat box and sa comment section po. So ngayon po we have 168 participants. So Sige, habang naghihintay po, ako na po muna yung mauna sa question. So, tanong ko lang po, if possible ba na i-store yung nanobiopesticides sa or i-store siya ng matagal? And kung pwede siyang i-store, gaano po katagal yung storage niya?
Ayun po, thank you for that question, Mr. Jasper. No? So, to answer the question, yes po. And one of yung application talaga ng encapsulation technology is to increase the stability of the product. So, for my product, ang na-test ko lang po is storage stability after 1 month. So, after 1 month, stable pa naman siya. So, medyo hindi ko na lang napagpatuloy because of the pandemic. But if pwede pa siyang ipagpatuloy for analysis at longer periods of time in order to see how long will be the product stable at different conditions po.
Thank you po. Ano po kaya yung temperature nung storage na?
Yung sa assay na ginawa ko po, that's room temperature and I think 54 degrees. That's a standard method for determining storage stability, which is required for application ng sa pesticide by the FPA.
Meron po tayong question from Zoom. So from Emerson J. Ibacara. Good morning po. Ask ko lang po if the nanopesticide technology was already tested in vivo test field test. Kung na-test na po, ano pong commodity? Salamat po.
Ayan po. So, yung particular ko sa thesis ko po, the enzyme-loaded nanoparticles, I was not able to test it sa in vivo. Kasi when dapat mag-start na yung screenhouse assay but pandemic happened. But yung thesis ko po is actually a part of a larger project funded by the USDP card. So, in the project, we use another product, another nanoparticle, using not enzyme but secondary metabolites also from PGPB. So that particular product was already tested in vivo sa field for Fusarium oxysporum causing fusarium wilts ng cucumber, tomato, and banana. So for banana, both FOC-TR1 and FOC-TR4. So yung TR4 in Davao.
Thank you po. From Harold Paniza po. Sir, just wanna confirm, your technology is considered as organic nanoparticles? Kasi meron po tayong classification like inorganic or the metallic nanoparticle and inorganic and organic like yung liposomes and dendrimers, etc. nanoparticles.
I think it will be considered organic in the sense of yung kasi yung wall material, the core is an enzyme which is organic, and the wall material for liposomes is phospholipid. For the carbohydrate nanoparticles, chitosan is also organic. So as a whole, the product I think would be considered organic nanoparticles po.
So from Vincent po, so hello po, yung nanopesticides po ba may specific na insects or pests na makokontrol or madadamay yung mga ibang insects tulad nung pollinators?
Ayun po. So in my study, I only looked into the effects of the nanobiopesticide on the target pathogen, which is yung fungi. But as I've said earlier, it's also important to determine kung anong effect ng product or the nanoparticles on non-target organisms, such as yung microorganisms in the soil where the nanoparticles will be applied, and also in insects, both insect pests and natural predators. So that will be for future studies na po for the product.
again po from Harold Paniza. So also if ever po in vivo application, ano po ang possible mode of application? Foliar or drench application, especially in the field na po.
For our, because yung project is concerned with the control of fusarium wilt and yung fungal pathogen is soil-borne. So yung tinitignan namin na mode of application is drenching method sa soil. So both preventive and curative applications. So yung syempre yung mode of application would depend dun sa characteristics of the pathogen that you want to control.
Ayun. Thank you po. So mukhang wala na pong kasunod na question. Meron pong kasunod na question from Harold Panisa uli. So also knowing na chitin enzyme ang target, it's also applicable sa insect pests, right? Sir, right? I think some insect exoskeleton ay made of chitin din po.
Yun po, tama po yun, Sir Harold. Since tama nga, chitin is also a part of the exoskeleton of insects and other arthropods, pwede siyang explore yung use of the chitinase nanoparticles in terms of control of the insect pest.
Yan. Ayun, so last question na po yata yun. So, ayun, salamat po Mr. Chris Lord for the lecture po and for also accepting our invitation po to speak in this seminar. So, salamat din po sa viewers natin for the participation. Binabalik ko na po sa ating MC ang ating programa, Miss Sarah.
Okay, so thank you again, Mr. Jasper, for moderating the open forum. And of course, to our speaker, Mr. Santos, and also to our audience. So we will now proceed to the awarding of certificates. As a token of appreciation to our guest speakers, we would like to present this certificate of appreciation to Dr. Lilia M. Fernando Corpus for serving as resource person on nanobiosensors for plant pathogen detection during the NCPC webinar titled Nanobiotechnology Applications for the Management of Crop Pests and Diseases, held on 27 September 2022 via Zoom, given this 27th day of September 2022 at the National Crop Protection Center, College of Agriculture and Food Science, University of the Philippines, Los Baños, Laguna, Philippines. Signed, Charlotte Hagosahos, our chair for the Seminar Series Committee, and Dr. Barbara L. Kawili, NCPC Director. Thank you, Ma'am Lilia.
Thank you, and congratulations to NCPC.
Thank you po ulit. Okay, again, a certificate of appreciation is awarded to Mr. Chris Lord T. Santos for serving as resource person on nanobiopesticides during the NCPC webinar titled Nanobiotechnology Applications for the Management of Crop Pests and Diseases, held on 27 September 2022 via Zoom, given this 27th day of September 2022 at the National Crop Protection Center, College of Agriculture and Food Science, University of the Philippines, Los Baños, Laguna, Philippines. Signed, Ms. Charlotte Hagosahos, Chair of the Seminar Series Committee, and Dr. Barbara L. Kawili, NCPC Director. Thank you again, Mr. Lord Chris Santos, and thank you for sharing with us your work and knowledge. Okay. To close our program, for those who are requesting for e-certificate, please do not forget to answer the evaluation form through the link presented here. E-certificates are given only after you answer the evaluation form. Okay, to close our program, may I invite Ms. April Alviar, the Deputy Director of NCPC, to give us the closing remark.
To our resource speakers, College of Agriculture and Food Science Associate Dean for Research and Extension, Dr. Lilia M. Hernando Corpus, and Center for Natural Sciences and Environmental Research of De La Salle University, Science Research Specialist, Mr. Chris Lorty Santos, and to all our attendees for today's webinar, good morning to all of you. I would like to express my appreciation to our resource speakers for accepting our invitation to be the speakers in today's webinar and for delivering a very informative, insightful webinar. Nanotechnology has provided new tools in the form of nanosensor and nanobiopesticide for pest and disease management in agriculture with the concept of minimal usage for maximum effects. Thank you, Ma'am Leah, for your talk about the nanobiosensor for plant pathogen detection. This has been a great opportunity for us to see the development of new rapid and sensitive detection and identification technologies for plant pathogen. High cost, difficult of complicated process, and specialized training on the conventional detection methods are some of the problems hindering the rapid diagnosis and detection of plant pathogens. Nanobiosensor offers a new view because of its high sensitivity and specificity and compatibility with data processing technologies. Mr. Santos discusses study about nanobiopesticides, which gain special attention again, pests and diseases due to high stability, easy field application, and cost effectiveness. It's also biopesticides also are environmentally friendly and less toxic. And these studies show potential for the application of this nanopesticide in controlling pests and diseases. Thus, nanotechnology is considered an important achievement in the agriculture section in order to improve pest and disease management for increasing yield, promoting pest and disease resistance, and advancing sustainability. Would like also to take this opportunity to commend the organizing committee for successfully conducting this webinar series, and thank you also to our attendees for actively participating in our activity. We hope to see everyone in our succeeding webinars. Maraming salamat po.
Thank you, Deputy Alviar. Once again, we thank all the participants of this webinar. If you wish to watch the video presentations for today, visit the Facebook page and YouTube channel of the National Crop Protection Center. Okay, muli, magandang araw po sa ating lahat and see you again in the next NCPC webinar series.